Our second speaker is Sarah Senese. Sarah hails from New York and graduated from Colorado College in 2023 with a bachelor's in organismal biology and ecology. Prior to her internship, she worked on physiology of cottonwoods and riparian ecosystems, and also had research experiences in Costa Rica, as well as a backcountry trip. The research project Sarah will be, the research project she'll be presenting on really gets at the heart of the uniqueness and specialness of our endemic scrub species. And that's how plants tolerate allelopathy, or essentially the chemical warfare between plants from this dominant Florida rosemary. So as you'll see in her talk, she combined laboratory assays and field surveys to gain these really cool, important insights into the various ways that species tolerate or even rely on allelopathy from the rosemary. Sarah is a true nature lover, and not only becoming an expert in the plants around here, but quickly learned the birds and insects that we have here as well. She self-describes as funny. And also has witty banter. Well, she can't look up anymore. And has this amazing encyclopedic knowledge of music. Sarah will be heading back to Colorado for the summer for an exciting opportunity to research deer mice with the University of Massachusetts. And then she'll be coming right back here to work as a research assistant with us this fall. So hand it over to Sarah.
Sure. Yeah, it's fine. I also have sound prepared for that, Laura. Yes, I do, but I have sound on the video. Yeah, if that matters. This way? Oh. It is a video clip. Okay, cool. It's really important to plates. Okay, great. Awesome. So yeah, as Erin mentioned, I will be giving my talk, Am I Special? Effects of Ceratiola ericoides allelopathy on endemic bald specialists and generalists. So my story in the scrub here begins in the rosemary bald. When I first came to Archbold, a lot of what the plant ecology program's projects were happened to be in rosemary balds. And this is unmistakably a really interesting habitat, regardless of the fact that it's an interesting scrub habitat. It's just plain old bizarre. We have these islands of Ceratiola ericoides, the Florida rosemary. They're all super isolated. They form these little mountains. And what really took me about this specific environment wasn't necessarily the rosemary and that way that they spread out, but was more the gaps in between them. And more than the gaps, the herbaceous plants that were occupying those gaps. Because to me, especially in an environment that's so mediated by burn and fire, I was wondering sort of who gets to be here? Why are these plants in the gaps as opposed to anywhere else in this habitat? And then sort of lastly, how does competition influence this landscape? So I began looking at this idea of competition and sort of digging more into the literature. And obviously the first hurdle here is that Ceratiola ericoides is gigantic and all of the herbaceous plants are very small. And that's the first competitive advantage that Florida rosemary has against anything else. But the second is allelopathy, which is when a plant exudes chemicals into the soil, which inhibits the growth of other plants around them. And that's well and great, but I'm less into a lot of the literature about competition, and I'm really into literature about facilitation. And this idea of allelopathy is great, it's cool, but it kind of leaves no room for facilitation, because if you go into a rosemary bald right now and you just walk around, there's obviously herbaceous plants all over the ground. So clearly something else is happening here that isn't just, plants fighting each other, duking it out and dying. So I began to think, how could facilitation play a role in this environment? And I thought maybe it's not while the Florida rosemary is alive and well and giving off allelic chemicals, but maybe it's after it dies. Maybe it's the absence of the allelic chemical that allows these herbaceous plants to germinate, and therefore it's sort of a delayed facilitation after the rosemary's death. So who does this sort of have to do with? We have our specialists and our generalists. There are plants that just naturally occur in rosemary bulbs as opposed to other places. And then there are generalists who can sort of hang out in a lot of different kinds of places. So I wanted to see how this idea sort of fit into the idea of allelopathy facilitation and this delayed facilitation, which brings us to the ghost nurse plant hypothesis. We were deciding, Erin and I, whether or not it was going to be ghost or zombie nurse plant. It was a very spirited discussion, but we landed on ghost nurse plant. And this is that Ceratiola ericoides is sheltering the seeds of these tolerance specialists, these allelopathic tolerance specialists, until the rosemary's death. And so in order for this hypothesis to be true, other hypotheses need to be true about the specialists themselves. First is that they need to be potentially dose tolerant. They have to be able to tolerate way higher doses of allelic chemical than other plants. They also need to therefore be able to tolerate longer periods of allelic chemical exposure. And then lastly, a hypothesis, and this was kind of farther of a reach, was that specialists might be delaying their germination to wait for the ghost nurse plant's facilitation. So waiting until the rosemary dies, a fire comes in order to strategically germinate and capitalize off of these gaps. So how do we characterize allelopathic tolerance? We did this 3 ways. First is through concentration. So how much allelic chemical can generalists and specialists just tolerate? Bar none. How much can they tolerate? How much can I dose them and see when they'll die? Then we have exposure. Who can tolerate full-time allelopathic pressure? And then lastly, this idea of delayed germination. Who is forming seed banks? And is there this allelopathy-mediated seed dormancy? So before we can really talk about anything that I did, we have to talk about the main player here, which is hydrosenamic acid. So Florida rosemary exudes a chemical called ceratiola. Ceratiola degrades in the soil into hydrosenamic acid through volatilization, root exudation, bleaching, and decomposition of plant materials. And what's really cool about hydrosenamic acid is that you can go on the internet and order it off of Thermo Fisher Scientific right here. It's easily soluble in water. And it also is in a lot of household products and foods. And those of you here should actually already know that if you had read The Scent of the Scrub, Florida Rosemary's Aromatic Secret, which can be found in the local secret section of the Highland Sun. So this shouldn't be new to anybody. But in all seriousness, it is really cool because I'm going to be talking about HCA, hydrosenamic acid, is this like deadly warfare allelochemical, when in reality, it's actually giving our scrub this super characteristic scent. And it's cool that it kind of plays both sides of the coin. But to get back to the science, the first experiment that I did in the lab after collecting all of my seeds was to look at that aspect of characterizing allelopathic tolerance through concentration. And what I used is a dose response experiment. What's cool about dose response experiments, especially in this context, is that it's pretty much exclusively reserved for culture. So it's used a lot of the time in dosing seeds with pesticides to see how much pesticide can be dosed before they die. And it's rarely done with allelochemical, and it's never been done here with allelochemicals, which is really cool. So my dose response experiment consisted of 5 treatments, my 0 parts per million, my control, that's just DI water. Then I have 15 parts per million, which is kind of not really an amount that you would find in the soil. So I wanted to go on the lower end. 50 parts per million, which sort of ticks up into where we would actually find the normal range of allelochemical in the soil. 500 parts per million, which is still in the normal range, but sort of leaving it. And then lastly, 1,000. So I wanted to get the 2 extremes and see who would germinate. I dosed them for 5 minutes in the beginning, and then I seeded them, watered them, and waited 8 weeks to see who would germinate. It required a lot of really tiny petri dishes and this really flattering photo that Erin took of me carrying a bucket of sand upstairs while everybody watched and nobody helped me after dropping it in the parking lot and making a mess. We also had to very closely monitor all of the plates. Here's our very own Sterling looking at my germinates with glasses that were never used after this moment. The second experiment that I did in the lab was sort of looking at the second two of my characterizing allelopathy. So that would be exposure and the delayed germination. And I did this with what we have dubbed the start-stop experiment. So all of my treatments received the same amount of allelochemical, 50 parts per million, and I had 4 treatments representing 4 different spans of exposure where acid was dosed on the seeds, as opposed to the dose experiment where they're dosed once for 5 minutes. In this case, I'm dosing them for 1 week, 2 weeks, 3 weeks, and 4 weeks. But I wanted to make this ecologically relevant. I wanted to see what would happen if we were simulating fire, simulating that ghost nurse plant. And so I replaced the acid after the 1, 2, 3, and 4 weeks, water. And so this was another experiment that went on 8 weeks, and these were my treatments. So in the front here, you can see my start-stop experiment. In the back is my dose response. Each of these rows is species. This is a treatment, so I have my 4 treatments, my DI water, and my 50 parts per million hydrocyanamic acid. This usually gets more laughs. Thank you. Okay. Thank you. That video is actually 10 minutes long, and the clouds move in the background to show that. So let's meet the species. Who are we working with? We have our generalists, Baldwina angustifolia, Leccia our allelopathy adapted specialists, which it's going to become clear later in my talk why I've split specialists into these 2 subcategories. We have Polygonum nasomii, Hypericum calicula, Leccia cernua, Liaetus olingeri, Orangium caenifolium, and our non-adapted specialist, Paronychia tartacea. So how do you characterize things into specialist or non-specialist? And that is with something called a specialization index, which I'm going to be referring to later in my graphs, which is just the proportion of the species occupying a rosemary bulb over the proportion in a rosemary bulb, and then some other comparative habitat, in this case, scrubby woods. So I've shown what I've done. Let's see what the results were. So for concentration, do generalists or specialists tolerate allelopathy? When you are doing a dose response experiment, the output of the fruits of your labor ends up being a dose response curve. This is an example of what one might get. Where on the y-axis, we have the response variable, the x-axis, the dose, and in this case, the response is survival. You want to find where on the y-axis you have 50% death. Go across, and that's where you find your LC50, your lethal concentration when 50% of the population dies. This is an example dose response curve I got for Baldwina angustifolia, one of my generalists. It might look different, but it's actually a really nice dose response curve, where if we go to about the half the germ total and go to the treatment on the x-axis, we find that the lethal concentration, 50%, is about 145 parts per million hydrocyanamic acid. We find that more specialized species can tolerate higher doses of allelochemical. So down here, we have our specialization index, with 1 being highly specialized and 0 being not specialized at all. And then we have my lethal concentration 50 dose. So down here, we have Baldwina angustifolia and Leccia. These are the generalists. This makes sense. They're low in the specialization index, and they can't really handle a lot of allelochemical. Then we have our weird friend Paronychia, who, according to all accounts, should be a specialist, for some reason really cannot handle allelochemical, which is why we're calling it sort of the non-adapted specialist. And then we have all of the adapted specialists who are sort of riding this trend of being able to tolerate anywhere from the normal range all the way up to where Liaetus is. Liaetus, you can do whatever you want to it, it'll still germinate, it wants to live so badly. So that's all the way up there in the corner. Next, we want to talk about exposure. So who can tolerate full-time allelopathic pressure? Specialists tolerate longer exposure than generalists. Again, we have our two generalists, Baldwina angustifolia, Lectioducardii, low on the specialization index, and they can only really take upwards of like a week and a half of acid treatment before they just putter out. We have all of our adapted specialists who can handle, anywhere from about 2.5 to an infinite number of crazy weeks that I wasn't able to actually do. Once again, Liatris. But if we revisit our weird non-adapted friend Paronychia, this time it's actually able to handle exposure, just not concentration in high doses, which is interesting. Next is sort of what's getting at the hypothesis of the ghost nurse plant, which is this delayed germination. And it might seem that some generalists are germinating in the acid treatment regardless, which would make you think, oh, they're fine. They actually have an incredibly low seed viability and are doing this sort of go for broke method. Baldwina angustifolia in any natural 0 parts per million condition germinates in about 4 weeks on average. So here in treatment 4, it seems like we have a lot of germination, but what's really happening is it's just normal growth trend, but with really low seed viability. So it's growing at the normal rate it should, but it's just spitting out a couple of seeds as opposed to if it was in 0 parts per million, where it would have, 100% germination. If we compare this with Hypericum cumulicula, which is a really good specialist, it's waiting until after the acid treatment, regardless of the dose, to germinate, which is really cool. Some specialists are waiting and delaying their germination until allelopathy is removed from a system. We have our two specialists, our non-adapted specialist, and our adapt specialist. Here we have percent germination in acid. In all of my species, you can see Baldwina angustifolia is sort of germinating in acid, but a lot of our friends, except for Liatris, who really wants to live, are sort of doing this wait and see strategy. But wait, Sarah, what about the generalist who appears to be delaying their germination until allelopathy is removed from the system? Because Lectioducardii really shouldn't be waiting if it's, not specialized and is doing this go for broke method. Lectioducardii seeds are really badly damaged by the acid and are just barely germinating it. Treatment 1, which is going to be the red line, has the most germination because it has the least allelochemical in the system. Treatment 3 and 4 don't even have a slope. They're just completely flat because if they are sort of getting some of these seeds out to germinate, it's happening in the treatments where the least amount of acid is in the system. If we compare this with, again, a really good specialist, Hypericum cumulicula, it is consistently germinating after acid has left the system. It's a high p-value and a high R-square, regardless of treatment 1 or treatment 4. So let's revisit the ghost nurse plant hypothesis. Specialists are potentially dose tolerant. Yes, they are. Specialists might therefore be able to tolerate longer periods of allelochemical. They can tolerate exposure. And lastly, specialists are delaying their germination, which is really cool. It's a point towards waiting for the ghost nurse plant. Well and good, but Sarah, are these predictions consistent with what we see in the field? So I went to 2 rosemary bulbs and I wanted to see if any of this was actually situationally relevant. So I went to 2 recently burned bulbs, 28 and 42. They have very similar sand conditions. And what's really cool about these bulbs is that they're on margins of other habitat that are not rosemary bulbs, because I wanted to be able to do a comparative study. So who associates with the ghost nurse plant? What I did was a 3-meter quadrat around a burned rosemary. And also I threw a flag in the air behind me, and wherever it landed ended up being the focal for my comparison, which was also a 3-meter quadrat. And I measured presence, abundance, and closest distances to that focal point, whether it was the middle of the burned rosemary or the flag that I threw down for all of the species. I found that the more specialized you are, the closer to the ghost nurse plant you can be. So on the x-axis, we have the average distance from the burned rosemary, and again, our specialization index. Our generalist friends are hanging out towards the bottom. They really cannot be close to the ghost nurse plant. Our non-adapted specialist is actually hanging out somewhere sort of near the generalists in this case. It's not really near the ghost nurse plant at all, but our adapted specialists are fine. They can be as close to this dead skeleton as possible. They do not care. We also found that dosage tolerance predicts the use of the ghost plant. So on both of these x-axes, I have the lethal concentration 50 for the hydrocyanamic acid dose. On the left here, I have the number of plants per meter squared, so the abundance. And over here, I have the average distance again from the burned rosemary. You can see our generalists are not plentiful. They cannot handle high dosages, so they are not plentiful. Paronychia seems to follow that trend as well, even though it's a specialist. Legudicrudii and Boldewine angustifolia, again, our generalists are far from the rosemary and they cannot handle high dosages. Same with the Paronychia. Specialists are more abundant and closer to the shadow of the ghost nurse plant than generalists. But again, Paronychia chartesi is remaining the weird, non-adapted specialist. And so what comes out of this? We have sort of two strategies that specialists and generalists are doing. There's the wait and thrive strategy, and then there's the go for broke strategy, or they just generally have really low seed viability, with our friend Paronychia in the middle being the non-adapted specialist, who's acting like a generalist in the sense that they do not tolerate allelochemical well, but they can delay dormancy and handle exposure like a specialist. And so what does this mean all in all when we look at the ghost nurse plant hypothesis? It's really that Ceratiola ericoides, but more specifically, the allelochemical it exudes, has the ability in its absence to facilitate growth of specialists. Generalists, not so much. It doesn't matter. Once there's allelochemical in the system, it does not fare well for them, except for the non-adapted specialist who is able to germinate, just has to be farther away from the ghost nurse plant where there's less allelochemical in the system. This is really cool because no one's really looked at nurse plants in the sense of like a post-death nurse plant. And this could be really critical to understanding the structure of these post-fire herbaceous plant communities in the scrub. It could dictate how often we burn, when we burn, what we burn, because we understand how the herbaceous, endemic, and possibly even endangered plant communities will grow back. Thank you.
All right. Thanks, Sarah. That was awesome. Sas, first question.
Yeah, that was a really cool talk. Thank you so much. I wanted to ask you, where do rosemary seedlings fall in that?
That is an awesome question. So in my original sort of architecture for the project, I wanted to look at how rosemary affects rosemary. So I thought that was really cool. Little did I know, rosemary actually is fruiting and seeding right now. So I was not able to include it in the study, but I did include it when I was measuring distance and abundance. And rosemary follows the same trend as the specialists. It can be really close to itself. It can hang out. And that's sort of how the progression of the rosemary scrub continues, but it grows much slower, which is really in favor of these smaller herbaceous plants to have their moment in the sun, add to the seed bank, die, and then sort of the cycle continues.
And this may sound dumb, but is the facilitation of those other tiny species in that like they're physically sheltered or something? Or is it that they are not susceptible to the chemicals that the generalists or the other species would be?
I mean, I wouldn't be surprised if there was some kind of physical aspect to it because the rosemary is so big, but more so it's that their dormant seeds can survive constant allelopathic pressure and have the ability to withstand that and then germinate in its absence. So when the allelochemical is eventually removed from the system, they can capitalize off of that, unlike the generalists whose seeds do not tolerate exposure or dosage well at all.
I was wondering if you read at all about how long the allelochemicals persist in the environment, like if the rosemary plants were removed either by disease or physically, and what you would predict for the species comp in that bald after the plants were removed, after the rosemary was removed.
Right. So it's really hard to categorize how long it stays in the system because it's so varied. I based my dose response concentrations off of papers that one Aaron David in the room was on, and a lot of the HCA concentration, it was like anywhere from 50 to 450. So it's really hard to even nail down how much is in the system at all. But the way that I could answer that is that I think it's a gradual. So I don't think it's, the rosemary dies and immediately it's from the system. I think there's already probably a distance gradient, which is in play. And then I think that distance gradient probably has some time component, which is adding to it, but that would be super cool.
Any more questions?
Incredible presentation. In habitats in these areas where fire regimes have been suppressed, are you seeing a lot less diversity and density of these specialists and generalists in the middle of these Rosemary Islands?
So I looked at 2 rosemary bulbs that were recently burned within 2 years, but in the same way that I can tell you anecdotally that Paronychia cherhacea is like everywhere on station, it's in fire lanes, it's like growing on the side of the building, it like does not care what's happening with it, which I think is why it's like a weird specialist. In the same way that I can answer that anecdotally, I can definitely tell you that when you are looking at rosemary bulbs that have not been burned in a while, it's like Leonia, heaven, there's just like oaks galore, it's crazy in there. And I would say that definitely all of those things further suppress the endemic endangered herbaceous plants.
So we have a question online from Fern. Would you expect to find ghost nurse plants in other study systems?
That is a really good question. I think that a lot of study systems that have allelochemical and allelopathic plants like creosote and other types would absolutely have this, post-death zombie ghost nurse plant. I also just think it's really interesting to sort of look at ideas of competition on the other side of the coin, because it's, you can go into any system and see different things growing at different times. And when you're talking about allelochemical, a lot of the times it's like with really harsh vocabulary, and especially with competition as well. And I just think that it's cool to look at these systems as possibly having some more of a harmonious, give and take. And I absolutely think that other systems would have these ghost nurse plants for sure.
All right. Well, we'll wrap it up there. Thank you all for coming. We have some refreshments outside, and let's do it.